Welcome back. In this video, I will talk about what are antibodies, antibodies structure, types of antibodies, and key functions of antibodies. Now, what are antibodies? Antibody is also known as an immunoglobulin. These are large Y-shaped blood proteins produced by plasma cells in response to the presence of a foreign substances called an antigen. Antibodies recognize and bind to antigens in order to remove them from the body. A wide range of substances are regarded by the body as antigens, including disease-causing organisms and toxic materials such as insect venom. Now talk about the structure of antibody. An antibody has a Y-shaped structure made up of 4 polypeptide subunits. Each subunit has 2 identical light and heavy chains. The end terminus of each heavy chain forms an antigen binding domain with a light chain. There are 2 antigen binding domains forming the arms of the Y-shape. They are known as fragment antigen binding or Fab domains. The C terminus of the heavy chains forms fragment crystallization or Fc domain, which helps in the interaction with the effector cells. All 4 polypeptide subunits are held together by disulfide and non-covalent bonds. The heavy chains of the antibodies contain a variable region and 3 constant regions. Each antibody has 2 identical antigen binding sites and they differ in the antibodies. Now types of antibodies and their functions. Antibodies are classified into isotypes, which are defined by individual type of heavy chain constant domain, each isotype being encoded by a separate constant domain gene. In humans, there are 5 heavy chain isotypes: gamma, alpha, mu, delta, epsilon, corresponding to 5 antibody isotypes: IgG, IgA, IgM, IgD, and IgE. Immunoglobulin G or IgG. The most predominant isotype found in the body is IgG. It has the longest serum half-life of all immunoglobulin isotypes. IgG is further subdivided into 4 subclasses based on structural, antigenic, and functional differences in the heavy chain constant regions. These are IgG1, IgG2, IgG3, and IgG4. The IgG subclasses exhibit different functional activities. For example, IgG1 and IgG3 antibodies are generally induced in response to protein antigens, whereas IgG2 and IgG4 are associated with polysaccharide antigens. It is transferred to the placenta through the fetus and protects the infant until its birth. The heavy chains of IgG have 2 antigen binding sites and are of subclass of gamma. It facilitates the process of phagocytosis and provides immunity to the developing fetus. It neutralizes the toxins and pathogens and offers protection to the body. Immunoglobulin N or IgM. IgM is the first antibody produced in response to a microbial attack by B cells. It is the largest antibody and is found in a pentameric form. It circulates in the blood and lymph and constitutes 6% of the total antibody content in the serum. It is involved in agglutination and opsonization. It has a large number of antigenic sites on its surface and therefore facilitates efficient activation of the immune system. The heavy chain of IgM are the subclass of mu. Immunoglobulin A or IgA. Usually found in liquids such as breast milk, serum, saliva, fluids of the intestine. IgA in breast milk protects an infant's gastrointestinal tract from microbial activity. It constitutes 13% of the total antibody content in the serum and is divided into 2 subclasses, IgA1 and IgA2. Among these, IgA1 is highly found in the secretions and is also called the secretory immunoglobulin. It exists in both monomeric as well as dimeric forms. It provides the first line of defense against the pathogens and limits inflammations. It also activates the complement pathway and participates in the immune response. The heavy chains of IgA are of the subclass alpha. Immunoglobulin D or IgD. It is involved in the production of the antibody by B cells. It is present as a monomer and weigh around 180,000 Dalton. It comprises less than 1% of the total antibody content in serum. It acts as a receptor on B cell surface and participates in B cell activation and differentiation. The heavy chains of IgD are of the subclass of delta. Immunoglobulin E or IgE. IgE is present in the least amount, around 0.02% of the antibody content in the serum. These are present in the lining of the respiratory and intestinal tract and response to allergic reactions. This is found as a monomer in the body and weigh about 2 lakh Dalton. The heavy chain of IgE are the subclass of epsilon. Now, key functions of immunoglobulin. The key functions of immunoglobulins are agglutination, precipitation and neutralization of antigens, lysis of antigens, opsonization, assist in phagocytosis, directly attacks viral pathogens. They also act as the first line of defense for mucosal surfaces, activates mast cells and basophils.
अगर आपको ये वीडियो इन्फॉर्मेटिव लगी हो तो हमारे चैनल को सब्सक्राइब करें, वीडियो को लाइक करें। धन्यवाद।